I need to share my screen. You should be able to see Cystic Fibrosis Mutant Project. Do you see that document?
I do see it.
Scott sees it. Okay, hopefully everybody does. All right, I won't go over these URLs or the list of mutants. What I would like to do is start with in the center here where it's Cystic Fibrosis Project. Now, I sent you all a list of the groups and the mutants that have been assigned. Do you have those? Has everybody seen what group they're in?
I have not seen that yet.
Okay, then I'm going to show that first. Hang on a minute.
Okay.
Bear with me, I have to find it. Can you see groups, members, mutants?
Yes.
Okay, so these are the groups. There are 10 for this section, and you should be able to find your name in one of the groups under members, and you should be able to see the mutant that's been assigned to your group. So as I said, this is in Canvas. And so you can look it up on your own, but here it is. So you can write down which group you're in and who your group members are and the mutant that you've been assigned. So now that you've all seen that, I'm going to stop sharing and go back to the other document. Now you should be able to see the cystic fibrosis document, and I'm going to go down. Here's a list of mutants. So your CF project, worth up to 50 points per student. One significant change that I've made since we talked about this the last time is that your presentation will be 8 minutes rather than 10. Then prepare your presentation to illustrate the following points: the protein structure of your mutant, the protein function of your mutant, where the function breaks down, the symptoms caused by your mutant, and possible treatments for people that might have your mutation. And then you will be graded according to adherence to the foregoing instructions and the quality of your slides. There's a link here that you can click into. And also at the end of this document, I've included some tips for slide making. There are 4 points here. One is pare down your content. You'll gather a lot of information, but pare it down to the bare bones, refine your message, and have one key message per slide. Don't try to say too much per slide. Simplify your statements. Use the fewest words possible to make the point. And that's done more easily when you turn sentences into bullet points. And if there is a small point you want to make, you can have it as a talking point, a supplement to your slide, and you don't necessarily have to show it on the slide itself. And then number 3 here is an example of how simplification can be done. The elements of style that you'll use include the layout, the way you lay out elements of your slide, the colors you use, and the fonts that you use. So here's an example of a marketing strategy. Huge market opportunity. It's written out in sentences. Well, that can all be simplified down to just a few bullet points. And then if you want to make it more simple and more eye-pleasing, those bullet points can be boiled down to a slide like this. So it's an example of how you can use color and add decorations to make your slides more readable. You can illustrate your points. So for example, here, luxury smart watches, show a picture of a watch. Wallets, show pictures of wallets, and shoes. And then sometimes you have to decide what looks best to your eye. So here are 2 alternate ways of representing those products. If one of them looks better to you, then that's the type you should choose. And finally, when you're done, your slide always looks better if you align things. And this is easily done in PowerPoint using the alignment tool, which is a pull-down menu. You pull it down and you can align both vertically and horizontally. And then to lock things into place, once they're aligned, you group them using the grouping feature that you'll find in PowerPoint. Okay, this document is in Canvas, so you can study it at your leisure. Now, the final point I want to make is that each presentation must be reviewed and approved prior to May 26th. May 26th is the proposed date when you're going to make your presentation to the class. So you're going to need to make an appointment with Scott. Scott is going to conduct the reviews, and you need to make an appointment with him, which you can talk about in a minute. And your presentation is going to be timed. So if you're over 8 minutes or under 8 minutes by a significant amount, points will be deducted. And if you fail to have your presentation reviewed in advance, 10 points will be automatically deducted on the top. So Scott, would you like to comment on how they go about making appointments with you?
Yes. So what will happen is May 10th, 12th, 17th, 19th, and 24th. Those are workshop days. The second hour, so from 3 to 4, will be dedicated to the slide reviews. What you will do is you will email me, and first come, first serve. We will set up 3 different appointment slots per each day, and whoever requests the first slot will get first choice at it. What you'll do is you'll come on in, you'll display your PowerPoint to me. Everybody should show me a slide that they've worked on, and then everybody should present that information on the slide they worked on, and then we will discuss a little bit, and I'll give you any recommendations if I have any, and let you move on out. So expect it will take about 20 minutes of your time or so during one of those periods. I'm going to post an announcement, so you don't have to remember it right this minute, but that is the plan.
So, excuse me, Professor.
Huh?
Garrett speaking. So, if we did the appointment with Scott, do we have to do a group meeting or individual?
This will be a group meeting with Scott.
Oh, so we have.
Yeah,
Yes.
you could have
So
your
all-
presentation together by the time you go for your review. And so yeah, all the members of your group should meet with Scott and have him review. As he said, each of you in your group should describe at least one slide, and each of you needs to make at least one slide because you need the experience of making a slide in PowerPoint. So once you've all done that, then you'll have an appointment with Scott to put it all, to show him that you've put it all together. Any other questions about how to proceed?
Yeah, I have a question. It's Marissa. So how exactly are we going to be presenting our presentation? Is it going to be just to you and Scott or like to the whole class?
It'll be the whole class. It'll be during the class period, tentatively on May 26th. And so instead of me expounding for an hour and 15 minutes, groups of you will take turns. So you'll have, we'll go down the list. The groups are numbered 1 to 10. Start with group 1. Group 1 will present in an 8-minute slot, and then we'll go to group 2 and go on. So there are 10 groups. That will be 80 minutes if we're very precise with time, which is 5 minutes more than a class period, but I think we can do that.
Oh, okay. Thank you.
I have a question. It's Ariana Watson.
Uh-huh.
Do we have to have, or is it like a certain amount?
A certain amount of what?
Slides.
Oh, that's up to you. You shouldn't have more than 8, but you don't have to have that many. You can have fewer than 8. And it's pretty much the number of slides should be a function of the points you're trying to make. So remember what you're trying to do. You're addressing 4 issues about your mutant: the protein structure, the protein function, the symptoms, and treatments. So you'll prepare slides to illustrate those. So for example, with protein structure, you may need 1 slide, you may need 2 or 3, depending on how complicated the issue is that you're trying to present. So you should get together, members of your group. You can agree on a time to meet. I've bolded here. All members of each group should be involved in gathering and organizing information. The presentation itself can involve 1, 2, or all 3 members of the group, and you decide that yourself within your group. And then a score after you present on the 26th, a score will be assigned to your presentation. And each member of your group will get that score. Whatever your presentation scores is what each member gets. And I'm suggesting that you communicate among yourselves by email, and also you might have group meetings with Zoom.
I also have one more question. It's Marissa.
Uh-huh.
Can you, is the the list of the groups, or is it published on Canvas
Yeah,
already? It is?
yeah. You should
Okay.
find it, well, on your home page, if you look under modules, it's there.
Okay, thank you.
Okay, anything else? And this document also,
I have a question.
yeah.
This is Lisha. So the way we're gonna be graded is by how we all do as a group, not just individual. So we all get one grade.
That's right.
Okay.
Yeah.
How exactly are we going to be doing the presentation itself? Like, are we going to be sharing our screens over Zoom, or are we going to like have some sort of like thing where everybody goes to like a website and that way we're able to do it that way? Or
We're going to have to share your screen. So whoever is the presenter or presenters, we're going to have to share the screen with the class. Now, that's a good point. How are we going to do that? I may have to set up a special Zoom session. Does anybody know off the top of your head how we can allow everybody in the class to have to be able to show their screen?
not off the top of my head, no.
Okay, I'm gonna have to look into that.
Well,
Professor,
I
Uh-huh.
can see like a green button that says share screen.
Okay, so if you click on that, can we see your screen?
Yeah, you
Yes.
should. Yeah, as
Yeah.
long as you allow access, I think any student can share their screen.
Yeah, you just have to allow them to share screen and that's it.
Yeah, I think that was one of the things that we got ironed out early on in the semester, Dr. Yoder. It's working this semester. It wasn't last semester.
Okay, all right. So I don't have to change any settings in Zoom in order
No, sir.
to show your screen. Okay, so then you decide within your group how to, who's going to share their screen and who's going to do the oral part.
Professor,
Uh-huh,
this is Mary Cat.
uh-huh.
Is it possible to change the selected group?
Say that again.
It is possible to change the selected group?
Is it possible to change the group?
Yeah.
Not unless you had a very compelling reason.
Oh, okay. Thank you.
Okay, anything else? Make sure you're clear on what the assignment is and how you're going to go about it. You can probably clarify that when you get together with other members of your group.
Also, Professor, is there a limit on how many images we could add, or is that not going to be an issue?
That's up to you. I would say try not to use more than 8 slides. You've only got 8 minutes, and to show the slide, have people assimilate what's on it, and for you to describe it, a slide a minute is about the maximum.
All right, thank you.
Okay. Anything else before I take this down? All right, and I'll stop sharing that.
Professor, I have a question. It's Sarah.
Uh-huh.
Will we have any other projects besides the one coming up in May?
No.
Okay.
No other projects. Now you will have worksheets. So worksheets is another way of studying, and it also, if it's a graded worksheet, it can earn you points.
Thank you.
Hey, Professor, I'm looking through Canvas for like the files and I cannot find any.
Are you looking on your home page? And under modules?
I'll check again, but I looked and I didn't find anything.
Has
I'm
anyone else?
able to see it under the week 10 module.
Okay, thank you.
For whatever reason, next week's module's up, so that might be why if you're looking at just the bottom one, that might be why.
Okay.
Yeah, that's right. I've got, what is it, week 11 module with almost nothing in it. There will be more. Are you seeing Mendelian genetics now?
Yes.
Okay. I don't want to go over everything that we did before, but I want to do a little bit of review just to emphasize some points and make sure that you're clear on the essentials here. So the first point I want to make is that the magnitude of the contribution that Mendel made. To reiterate what I said before, prior to Mendel, when people would try to study inheritance, they would take two parents of anything and mate them and then look at the progeny. And what they were really studying is they were trying to study polygenic inheritance. In other words, they were looking at segregation of many genes because their parents were not homozygous. They didn't know that, but that's what explains why they got this uninterpretable mixture of phenotypes when they would make a cross. So Mendel solved that by, before he did any crosses, he did inbreeding. And so that's a critical thing, critical insight that he had that no one prior to him had had. And the second thing he did was, after doing his inbreds and making a cross, observing what he saw in the progeny, then he took those progeny and crossed them to each other to get what's called the F2 generation. And that's what led him to understand the segregation of genes. So remember those two things that Mendel did that were critical for him to be able to make an advancement in discovering genes. So let me go down here to, yeah, so key step one is making inbreds. And I won't go through this slide in detail, but he was working with peas. And in order to make a cross, he had to dissect the flowers. He had to take the pollen out of a flower and put pollen from a different flower on that flower, and then close the flower back up and let seeds develop. So that was a very tedious process. And key step two was the F2 generation. So in the simplest thing, and I think this was the first experiment that Mendel reported, he took a plant with purple flowers that was inbred. So he did a crossing of progeny to progeny for probably about 6 generations, and always chose the purple flowers in the case of if he was looking at the purple trait, or white flowers if he was looking at the white trait. And so both of the parents were highly inbred. So all of the loci in each of them were homozygous. Now, when he crossed these, instead of getting a smear of some flowers more purple, some flowers less purple, some flowers shades of white, instead of getting a smear of that, he got all exactly the same thing. same color purple flowers. No whites. Then he took 2 of these purple flowered progeny and crossed them to each other. That's how he generated his F2, in which case, to his and everybody else's amazement, white reappeared. And it turned out that there was 1 white for every 3 purples. And they were clear, discrete classes. There was no ambiguity about the phenotypes. So that's step 2. He went to the F2 generation. We went over this before, but let me do it again to help you understand how you decide on on making a prediction about progeny. So in the case of this so-called monohybrid cross, a monohybrid means you're looking at just one trait. That's flower color. And so we know that P purple is dominant, and we know that the purple parent is inbred, so it's homozygous. And so we can assign it a genotype of big P, big P. Now the white plant, we know that white is recessive, and it also is inbred. So all loci are homozygous, including the flower color locus. And so we call it little p, little p. And if you look at the gametes produced by these 2 parents, the purple plant can only make big P gametes. And the white plant can make only little p gametes. And so if you mate a big P with a little p, what you get are all heterozygotes. Big P, little p. Those are the F1 plants. Now they can make gametes. And when they do, now half the gametes are going to get the big P allele, and half are going to get the little p allele. So that if you're crossing them at random, a sperm that's big P can fertilize an egg that's big P, or a little p sperm can fertilize either an egg with either big P or little p, or a little p can fertilize a little p. And when you do that, you get what Mendel observed. That is that the phenotype, 3 purples and 1 white, but the genotype is 1 homozygous big P, 2 heterozygous, and 1 homozygous little p. And that's the only one that's white. So is there any question about how this goes? And we talked about the definition of an allele. It's alternate forms of the same gene on homologous chromosomes. So these 2 purple chromosomes are homologs. On one of them, there's an allele for purple flowers, and on the other, there's the alternate allele for white flowers. So even though these chromosomes are homologous, they're heterozygous at this locus for flower color. Now, how did Mendel define dominant versus recessive? Well, the characteristic that appeared in the phenotype of the F1 was dominant. And there was only 1 characteristic that appeared in the F1 in any of the crosses that he did. The characteristic that disappeared in the F1 and then reappeared in the F2, that was what Mendel called a recessive character. And so this compares dominance and recessiveness on two homologous chromosomes. So at the P locus, the red chromosome has a big P and the blue chromosome has a big P. So this is homozygous for the dominant allele big P. At the A locus, both chromosomes have a little a allele. So that's homozygous recessive. And at the B locus, one chromosome has a big B dominant allele, one has a little b recessive allele. So this, at this locus, B is heterozygous, big B, little b. Genotype versus phenotype. Genotypes here on the left, big P, big P, big P, little p, and then little p. Any plant that has a big P allele is going to be purple. Only if both alleles at the P locus are recessive do you get white. Okay, so let's go through an example of a monohybrid cross. So what we have is a true breeding tall plant. True breeding means that it's been inbred and is homozygous at all loci. And it's crossed with a true breeding short plant. Tall is dominant. What will be the genotypes and the phenotypes of the F1 offspring? And what are about the genotypes and phenotypes of the F2s? Well, we can go through that in the following way. So we know that the tall plant is true breeding and that tall is dominant. So we can assign it the genotype, the symbols, big T, big T. So if it makes gametes, all the gametes are going to be big T. The short plant is also true breeding, and short is recessive. And so we assign it the genotype little t, little t. The only type of gametes that it can make are little t. So if you cross these two, the F1 genotype is going to be a heterozygote, big T, little t. Because tall is dominant, all of the F1s are going to be tall. But now when these heterozygotes now, among the F1 generation, you can pick two individuals from that progeny and cross them. And when you do, the gametes that they produce can be either big T or little t. And so what we're doing is crossing two F1s, which are both heterozygotes, so that if you look at this little table here, either parent can contribute either a big T or a little t. Parent 1 can do that. Parent 2 can do that. So if a big T sperm fertilizes a big T egg, you're going to have a homozygous dominant big T little, big T big T. If it's little t and big T, then you're going to get heterozygotes. And if it's little t, then a homozygous recessive. So to answer the question from the previous slide, what are the genotypes and phenotypes of the F1? Well, the genotype is big T, little t. The phenotype is tall. What are the genotypes and phenotypes of the F2s? The phenotypes are 3 tall. So anything that has a big T allele is going to be tall because that's dominant. 3 tall to 1 short. The genotypes will be 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. So is that clear how you go through this? Because you do have a big problem set where you're going to go through this several different times with different kinds of genetic parents. And so you're going to need to know how to go through this process. Now, something else you're going to need to know is what is a test cross and how to do one, and why would you do one? And make sure you know this, because I'm sure we're going to ask it on a test at some point. So the situation is that you have a phenotype, in this case, purple. And this purple flower can be either homozygous dominant or a heterozygote. You don't know which. But you also have a white flower. Now you know what its phenotype and genotype are. The phenotype is white, and you know that if it's white, it's homozygous recessive because it's an inbred. So you know one parent, you don't know the genotype of the other, so you cross them. So if this purple plant is big P, in other words, homozygous dominant, then all the progeny are going to be purple. As you can see that here, that every gamete is going to be big P, and so regardless of whether there's a little p there or not, there's always a big P there, and all the progeny are purple. If, on the other hand, this original purple parent is heterozygous, then you're going to be crossing a little p with a big P little p. You can see that here in the Punnett square. So half the progeny will be purple and half will be white. If that is the outcome, then you know that the original purple parent was heterozygous. Okay, so you need to know what a test cross is and why you would use one and how to do it. The key is to use one parent as a homozygous recessive. You know the genotype of that. So then what happens if you're trying to follow more than one characteristic? And if it turns out you're trying to follow 2, that's called a dihybrid cross. So in this example, it's taking a parent that has smooth seeds that are yellow, 2 different traits, and you cross it with a plant that has wrinkled seeds that are green. So in the F1 generation, all the plants are smooth and yellow. So that means that smooth is dominant, that's big S, and yellow is dominant, that's big Y, big Y. And if you make crosses among the F1s to get an F2 generation, now the segregation is going to be 9 smooth yellows, 3 wrinkled yellows, 3 smooth greens, and 1 wrinkled green. And you can follow that in the following way. So what's important that you do when you're doing these problems is always ask yourself, what are the gametes? So in the first cross, in the parental generation, the gametes are all big S, big Y for one parent, and little s, little y for the other parent. So the F1 generation then is going to be 100% heterozygous. Big S, big Y, little y. So if you make your Punnett square with these genotypes plotted on the two axes, you have big SY, big S, big Y, big S, little s, big Y, little s, little y on both axes. And so if you plot these out, you can determine what colors and what textures each of the seeds is going to be. And so if you count these up, there are 9 smooth yellows. You count the green ones, the smooth green ones, there are 3. If you look at the wrinkled yellow ones, there are 3, and only 1 wrinkled green. So is that clear? Because you're going to be doing this yourselves when you solve some of the problems on the worksheet.
I have a quick question.
Yeah.
So, assuming we know how many total alleles there are in the human genome, has anybody ever done the calculations to try to find out how many different combinations there could be of people?
I don't know if anyone has ever come up with an answer, but it's it would be almost infinite, considering that you have roughly 20 to 25,000 genes, protein coding genes in the human genome. Yeah, the number would be just astronomical. Scott, do you have a comment about that?
No, it's an interesting question, but again, I think you're probably starting to talk numbers that are so large they're meaningless.
Probably. I was just wondering.
Well, yeah, that's there's the general conclusion that no two humans are alike, and that's why there are just so many possibilities, considering that all the different allele choices. And it also makes the point, we don't have inbred humans. So whenever you make a cross between two humans, it's heterozygous at many different loci. So if you look at the children of any two parents, they're going to be vastly different unless they're twins. Usually they're vastly different from one another.
And even if they're twins, and they might be identical twins, they don't even behave the same in a lot of cases. So there are differences somewhere in there. You know, the chances of finding two people that are exactly the same are infinitesimal.
Yeah, and one of the things that we're not going to teach you about in this course is epigenetics. So even as Scott says, if there are so-called identical twins, as soon as they start to develop, there's going to be epigenetic changes that causes them to have different phenotypes. And the older they get, the greater the differences in their phenotypes.
All right, thank you.
Okay. Now, here's something else that you need to know. Mendel's laws of inheritance: segregation, independent assortment, and dominance. So these are the conclusions that Mendel himself made from studying the results of his crosses. So segregation is when allele pairs separate during gamete formation and then randomly unite at fertilization. And I've copied here a picture from our cell division lecture where you see the segregation. These are the gametes on the right, and you can see that each gamete is unique because the chromosomes have segregated. So that's the law of segregation. the law of independent assortment, chromosomes separate randomly from one another at metaphase. And if you remember this picture from the cell division lecture, the chromosomes line up in metaphase in the center of the cell, and they have to go one way or the other, to one side of the cell or the other. And which side they go to is entirely random. So that's called independent assortment, or sometimes random assortment. And his final law came from his analysis of the F2. The dominant allele completely masks the effect of a recessive allele. But the dominant allele produces the same phenotype in heterozygotes, and homozygotes. So if it's homozygous dominant, you see the same phenotype as if it's heterozygous. Now there can be variations in levels of dominance. So as we've just said, dominance indicates that an allele masks the phenotype of its alternate. Now, codominance can happen for some traits. In the case of codominance, the F1 generation has both parental phenotypes. So if you're looking to study uniparental traits, codominance violates what you're looking for. Something else that can happen is incomplete dominance, where the heterozygote, in other words, the F1, the phenotype is a blend of both of the parental phenotypes. And then there's the exceptional case if a gene is on one of the sex chromosomes that changes the inheritance pattern. And we'll talk about that. So let's look at the case of incomplete dominance. Now you cross a red flower and a white flower. And this is actually true in the case of chrysanthemums. Cross red and white, the F1 is a heterozygote, but instead of being either all red or all white, it's pink. And then if you go to the F2, if you cross two of the F1s, what you get is a 1 to 2 to 1 ratio, 1 red, 2 pinks, and 1 white, which is a reflection of codominance. Now, interestingly, Mendel was either very lucky or very crafty, because if you go back and look at the 7 traits that he chose to study, every one of them is dominant only. He has no codominance, no incomplete dominance. So people suspect that in all cases where he didn't get nice, clean segregation of single traits, he ignored those and only reported the results that he could interpret. Another case of incomplete dominance. This is in the case of a human disease, hypercholesterolemia. In the homozygous condition, you get these LDL receptors in the cell. The heterozygotes have LDL receptors, but only a few of them, and so there's mild disease. And only in the homozygous recessive, there are no LDL receptors and there's severe disease. So it's another case of incomplete dominance. So now let's look at sex-linked inheritance. So both males and females have 44 autosomes. and 2 sex chromosomes, XY in males, XX in females. So when a male makes gametes, half of his gametes are going to be X and half will be Y. When a female makes gametes, they're all X. And this is how we get half boys and half girls in the population, because at random, if an egg, which will be X, is fertilized by a sperm that is X, that's going to be an XX female. On the other hand, if an egg, which is X, is fertilized by a sperm that's Y, that's going to be an XY male. So you can get different outcomes depending on the genotypes that you're looking at. So if we consider the case on the left, where the female has a gene on the X chromosome, it's dominant. It's labeled big A. So both of the X chromosomes have a big A allele. The male has an X and a Y, and on his X, he has the alternate allele, little a. So when sperm fertilizes eggs, you can have two possible outcomes. Either in the daughters get either a big A and a little a, well, all the daughters get a big A and a little a, whereas the sons get a big A and a Y. So all the progeny have the big A allele when the starting parents have these genotypes. On the other hand, supposing that the female is heterozygous at A and the male has the big A allele, then there are 4 possible outcomes. And 3/4 of the progeny are big A and 1/4 are little a. And we don't know what trait the A locus is controlling. All we can say is these are the genotypes. And on the right, if you have yet another configuration where the female is big A, little a, and the male has a little a on his X, then the progeny work out to be half big A and half little a. So to make that real, we can say what the trait on the sex chromosome controls. And color blindness is one example. So red-green color blindness in humans is an X-linked trait. Now, males need only 1 copy of the X mutation to be colorblind. Females, on the other hand, have to have a mutation on both of their X chromosomes in order to be colorblind. So consequently, fewer females are colorblind than males. So here is an example of, could be a real world mating. A male who is colorblind has children with a woman who's a carrier for color blindness. What is the likelihood that their children will be colorblind? Now, this is typical of an example that you might run into on your worksheet. So let's go through it. We have a colorblind male who has he has 1 X and it's mutant. So we call that XB for blind. And he has 1 Y. So the gametes that he can produce are XB and Y. The female is a carrier, so we know that she has 1 normal X and 1 mutant X, XB. So the gametes that she can produce are X and XB. So if you plot those out, the male parent can contribute gametes XB and Y. The female can contribute XB, excuse me, XB and X. So what do you get? You get XB, XB. That will be a female, and she will be colorblind because both of her X's have a mutation. There can be an X with an XB. That's another female because it's two X's. But this one will be normal because only one X has a mutation, and a female needs two mutations, one on each X, in order to be colorblind. Now, you can have an XY, this X and this Y. Excuse me, this XB and this Y, the child is XBY. This is going to be a colorblind male because a male only needs one mutation on his X in order to be colorblind. If X fertilizes Y, you get an XY individual, which is male, and it will be normal because there's no mutation on the X. So the answer to the question, what is the likelihood that their children will be colorblind? Well, it's 50%. Half the children are likely to be colorblind, half the females are colorblind, and half the males are colorblind. So is that clear how you come to that conclusion?
Yes, it's clear.
Okay, good. Then what if you're looking at two traits? A true breeding tall purple pea plant is crossed with a true breeding short white pea plant. Purple and tall are dominant. Now, what are the genotype and phenotype of the F1? And if two of the F1 progeny are crossed with each other, what will be the phenotypes and genotypes of the F2 offspring? Well, that's something for you to work out. I've started it here for you by sorting out what are the gametes. So we know that the tall purple plant is true breeding, and the short white plant is also true breeding. And both purple and tall are dominant. So we can say that the tall purple plant is big T, big T, big P, little p. The gametes are all big T and big P. The short white parent is homozygous recessive, little t, little t, little p, little p. The gametes are all little t, little p. So the F1 genotype is heterozygous at both loci. And so the phenotype will be all tall and all purple. The gametes now that the F1 can produce are four types, which you see here. And so if you plot those out on two axes, a so-called Punnett square, you can work that out for yourself and see what ratios of genotypes and phenotypes you get in the F2. So that's homework for you. Then I've listed a bunch of terms. In order to, well, the first thing I wanted to emphasize here on this summary is to remind you to remember what the keys to Mendel's success were: inbreeding and going to an F2 generation. Then I've listed a number of words that you need to know the definitions of, because that's the language of genetics. And if you want to discuss genetics, you have to know the language. I won't go through these. You can this is intended as a study guide for you. And I think, yeah, that's the end. There are more slides after that that I don't want to discuss now, but rather, I think I left you with quite enough. So you have a worksheet with, I think, 10 different questions that you have to solve in much the same way that we've just gone through now. Those are due this weekend, and we're going to discuss them in class. They will not be graded, but we'll discuss how you do each of those, or at least selected ones of those, in class on Monday. So you should be able to find the worksheet with the questions in Canvas. Okay, anything before we end the session?
Yes, Professor, where is the worksheet? I looked at the assignment, I didn't see it.
Well, again, it should be in module, I think it's module 10 in your homepage. Have you looked there? Has anybody been able to find the worksheet?
Is it the Mendelian genetics practice problems?
Yeah.
It's in the next week's module.
Yeah, it's not this week's module. It's next, because we'll be discussing it next week. Okay, anything else?
Do we have a quiz this week? I wasn't seeing any.
No, the next quiz is next Wednesday, the 14th.
Gotcha, thank you.
And that will cover, let's see, that will cover cell division and Mendelian inheritance. So you will have on that quiz some problems to solve. Okay, anything else before we go? Okay, then I'm going to stop sharing and